Structure of Microsporangium. In a transverse section, a typical microsporangium appears near circular in outline. It is generally surrounded by 4 wall layers: the epidermis, endothelium, middle layer, and the tapetum. The outer 3 wall layer perform the function of protection and helps in descent of anther to release the pollen. The innermost wall layer is the tapetum. It nourishes the developing pollen grain. Cells of the tapetum possesses dense cytoplasm and generally have more than one nucleus. When the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissues occupies the center of each microsporangium. Microsporogenesis. As the anther develops, the cells of the sporogenous tissues undergo meiotic division to form microspore tetrads. Connective tissue: epidermis, endothelium, sporogenous tissues, middle layer, tapetum. Epidermis, endothelium, middle layer, tapetum. Microspore mother cell. Pollen grains. As each cells of the sporogenous tissue is capable of giving rise to a microspore tetrad, each one is a potential pollen or microspore mother cell. The process of formation of microspores from a pollen mother cell through meiosis is called microsporogenesis. The microspores, as they are formed, are arranged in a cluster of 4 cells, the microspore tetrad. As the anther mature and dehydrate, the microspore dissociates from each other and develops into pollen grains. Inside each microsporangium, several thousands of microspores or pollen grains are formed that are released with the descent of anther. Pollen grain. The pollen grains represent the male gametophytes. If you touch the opened anthers of hibiscus or any other flower, you would find deposition of yellowish powdery pollen grains on your fingers. Sprinkle these grains on a drop of water taken on a glass slide and observe under a microscope. You will really be amazed at the variety of architecture, size, shapes, colors, and designs seen on the pollen grains from different species. Scanning electron micrographs of a few pollen grains. Pollen grains are generally spherical, measuring about 25 to 50 micrometers in diameter. It has a prominent 2-layered wall. The hard outer layer, called the exine, is made up of sporopollenin, which is one of the most resistant organic material known. It can withstand high temperatures and strong acids and alkali. No enzymes that degrades sporopollenin is so far known. Pollen grains exine has prominent apertures called germ pores, where sporopollenin is absent. Pollen grains are well preserved as fossils because of the presence of sporopollenin. The exine exhibits a fascinating array of patterns and designs. The inner wall of the pollen grain is called the indent. It is a thin, continuous layer made up of cellulose and pectin. The cytoplasm of pollen grain is surrounded by a plasma membrane. When the pollen grain is mature, it contains 2 cells, the vegetative cell and generative cell. Vegetative cell is bigger and has abundant of food reserve and a large irregular shaped nucleus. The generative cell is small and floats in the cytoplasm of vegetative cell. It is spindle shaped with a dense cytoplasm and a nucleus. In over 60% of angiosperms, pollen grains are shed at 2-celled stages. In the remaining species, the generative cell divides mitotically to give rise to the 2 male gametes before pollen grains are shed. 3-celled stage. Vacuoles, nucleus, asymmetric spindle, vegetative cell, generative cell. Pollen grains of many species cause severe allergies and bronchial afflictions in some people, often leading to chronic respiratory disorders, asthma, bronchitis, etc. It may be mentioned that Parthenium or carrot grass that came into India as a contaminant with imported wheat has become ubiquitous in occurrence and cause pollen allergy. Pollen grains are rich in nutrients. It has become a fashion in recent years to use pollen tablets as food supplements. In Western countries, a large number of pollen products in the form of tablets and syrups are available in the market. Pollen consumption has been claimed to increase the performance of athletes and racehorses. When once they are shed, pollen grains have to land on the stigma before they lose viability. If they have to bring about fertilization, how long do you think the pollen grains retain viability? The period of which pollen grains remain viable is highly variable. To some extent, depends on the prevailing temperature and humidity. In some cereals such as rice and wheat, pollen grains are viable within 30 minutes of their release, and in some members of Rosaceae, Leguminosae, and Solanaceae, they maintain viability for months. You may have heard of storing semen or sperms of many animals, including humans, for artificial insemination. It is possible to store pollen grains of a large number of species for years in liquid nitrogen minus 96 degrees Celsius. Such stored pollen can be used as pollen banks, similar to seed banks in crop breeding programs.